Hi, my name is Fernando Ramirez, and I'm delighted for this opportunity to share work I'm conducting at NIMH under the supervision of Peter Bandettini, entitled 7 Tesla Volt fMRI of human V1 reveals an orientation tuned signal consistent with a columnar organization. In the year 2005, 2 fMRI studies were published back-to-back showing it is possible to decode the orientation of visually presented gratings from distributed fMRI patterns measured from human primary visual cortex. However, today, the nature of the underlying source of grating orientation information remains debated. 2 key hypotheses have been proposed to answer this question. The bias sampling hypothesis proposes that grating orientation information originates from the bias sampling of cortical columns by different fMRI voxels. As shown to the right, idiosyncratic voxel responses result from slightly different proportions of orientation to neurons being sampled by different voxels. By pooling such small but systematic orientation signals across voxels, a multivariate decoder might correctly classify the orientation of a visually presented grating. An alternative hypothesis, termed the edge effect hypothesis, is based on the observation that grating orientation can in principle be readily decoded from a perfectly balanced representation. This is one where no orientation biases exist across voxels. Note that voxels in the schematic representation shown to the right have exactly the same proportion of neurons tuned to each orientation, unlike the original version of the figure by Boynton to illustrate biased sampling and shown to the left. According to the edge effect hypothesis, interactions between the orientation of a grating and the border of the aperture used to present it may be sufficient to explain the decoding of grating orientation from fMRI measurements. This concept may seem confusing at first, so I will try to explain it a bit further. Imagine you are presented with the oriented grating shown on the screen. If all voxels were perfectly balanced with respect to their content of orientation to neurons, you may expect that all voxels would equally respond to any grating, and hence grating orientation could not be decoded from fMRI patterns. The cool thing is that this is not the case. Edge effects due to interactions between the orientation of the grating and the border of the aperture used to present it lead to potentially decodable orientation signals even in a perfectly balanced representation. Due to the way Fourier energy spreads, voxels with receptive fields at the top and bottom of the stimulus would respond more vigorously than other locations, leading to a coarse-scale map informative regarding the orientation of the presented grating. For this 45-degree grating, voxels in the top right and bottom left corners might respond more vigorously, and so on. Whether biased sampling or edge effects are the main source of grating orientation information in fMRI remains debated. We wanted to look into this issue a little bit further. Thus, we aim to assess whether the multivariate representation of grating orientation in human V1 is best explained by bias sampling or edge effects with a combination of ultra-high field fMRI, pattern analysis, and synthetically generated stimuli. While we had no strong predictions regarding which hypothesis might best explain the data, based on the available evidence, we expected some contribution from each mechanism. Moreover, due to smaller voxel sizing or compared with previous studies, we expected a robust influence of the orientation signal attributed to bias sampling. To distinguish the 2 hypotheses, we generated flower-shaped stimuli such as these. Each flower was defined by one combination of grating orientation, 8 levels, radial frequency phase of the flower-shaped apertures, 4 levels, and stimulus contrast, 3 levels. We presented 6 subjects, each of these 96 flower-like stimuli, and measured bulb responses in primary visual cortex with 1.2 millimeter isotropic resolution. We selected visually activated voxels in V1 and conducted pattern analysis to assess the representation of grating orientation within and across radial frequencies. As explained next, the biased sampling and edge effect models make distinct predictions regarding the influence of aperture phase on the representation of grating orientation, and will allow us to distinguish these 2 hypotheses. For simplicity, we will ignore for now the contrast manipulation in our experiment. This will lead to more digestible 32 by 32 dissimilarity matrices, like the one shown here, instead of 96 by 96. This matrix shows all pairwise dissimilarities of a flower-like stimuli according to a model instantiating the bias sampling mechanism. Throughout the talk, we will always rely on the correlation distance as measure of pattern dissimilarity. Each of the 8x8 blocks highlighted in this matrix captures the expected dissimilarity structure for our 8 grading orientations for a specific pair of radial frequency phases. For example, each 8x8 block along the main diagonal of the big 32x32 matrix shown here exhibits pattern dissimilarities when gratings are presented with apertures with the same radial frequency phase. For this in-phase condition, the bias sampling model predicts the representation of grating orientation should reflect grating angular disparities, and gratings in the same orientation should reveal minimal distances, as indicated by the blue cells along the main matrix diagonal. Instead, orthogonal gratings are maximally dissimilar, as indicated by hot colors. Importantly, the bias sampling model predicts the same representation of grating orientation also when the modulators of the visually presented gratings are in antiphase. In contrast to the bias sampling model, the edge effect model predicts that the representation of grating orientation for gratings presented with apertures in antiphase will exhibit a very different dissimilarity structure. Perhaps counterintuitively, the edge effect model predicts that identically oriented gratings across apertures in antiphase will be maximally dissimilar. This is the opposite pattern of results as the bias sampling model. To summarize, let's arrange these key observations in a 2x2 table. While the bias sampling model predicts the same representational structure reflecting angular disparity within and across radial frequency phases, in stark contrast, the edge effect model predicts that gratings with the same orientation will be maximally dissimilar in the antiphase condition, as revealed by yellow cells along the main matrix diagonal. This leads to straightforward predictions when conducting RSA analysis with a model template reflecting grating angular disparities. While the bias sampling and edge effect models predict the same dissimilarity structure for gratings shown within apertures with the same phase, they predict opposite dissimilarity structures in the antiphase condition. Here, you see the average empirical dissimilarity matrix associated with grating orientation for the in-phase condition. It was computed across subjects. Although this condition is not informative regarding our key question, because both competing models predict the same dissimilarity structure, it is reassuring to see a clear agreement between the data and both competing models. Now, let's uncover the results of the critical antiphase condition. Even to the bare eye, the antiphase condition looks very much like the in-phase condition. The observed dissimilarity structure is inconsistent with the edge effect model. In contrast, the observed dissimilarity structure correlates with the bias sampling model. As noted when inspecting the main matrix diagonal, distances are close to minimal for both the empirical data and the bias sampling model. The observed pattern of results is summarized by the bar plot shown to the very right. Both the in-phase and antiphase conditions positively correlate with the model template reflecting grating angular disparity, which is consistent with the biased sampling model, but inconsistent with the edge effect model. It may be fairly argued that perhaps the pattern of results in our sample of subjects is heterogeneous or driven by one single subject. To rule out this possibility, we conducted RSA analysis separately treating the data for each subject. For completeness, we also show the two remaining radial frequency phase combinations allowed by our design. This is conditions when the phase over the apertures are dephased by half a cycle. On the y-axis of this bar plot, you see the mean and standard error of representational similarity analysis between the data and the usual model template reflecting angular disparity. As can be observed, the results are consistent across subjects and phase offset of the flower-like modulators. Now, we will get back to the stimulus contrast manipulation ignored thus far to simplify the presentation of our results. Neurons in V1 are known to exhibit not only orientation tuning, but contrast invariance. This is, V1 neurons maximally respond to the same grating orientation regardless of the contrast of the gratings. To test if the orientation-tuned signal we observed is also tolerant to stimulus contrast, we conducted an additional RSA analysis where we sorted all 8x8 matrix subblocks associated with grating orientation by the difference in contrast level of the visual stimuli. This bar plot summarizes our results. Mean correlations with the orientation model template in the y-axis are shown as a function of the difference in contrast level of the stimuli shown along the x-axis. The representation of grating orientation was found to be consistent across subjects and contrast levels. To summarize, we generated synthetic stimuli to distinguish the biased sampling and edge effect hypothesis of grating orientation decoding. The data only supported the bias sampling model. And finally, the identified orientation-tuned representation proved tolerant to changes in stimulus contrast, consistent with known properties of V1 neurons. Thank you for your attention.